All right, humoral immunity. I gotta warn you, this video probably gonna get a little lengthy. So just because there's a truckload of stuff to talk about related to this. Whereas the cell-mediated immunity was a lot more straightforward. Probably the most confusing part of that was just kind of the activation process and the role of T helper cells and whatnot. But with humoral immunity, I mean, I think this is a little more straightforward. But again, there's usually a greater volume of information related to this than there is with cell-mediated immunity. And my dingus cat just knocked over my notes. Thank you. Now, humoral immunity is a process that is carried out by B lymphocytes or B cells. Again, I'm just going to be referring to these as B cells as I go throughout this presentation. And now, remember that the main purpose of this process is to secrete, is to manufacture and secrete specific antibodies that carry out indirect attacks against foreign invaders, more specifically, foreign antigens. Now remember that concept of humoral immunity. Remember what a humor is, a body humor. It's just a body fluid compartment. All right, so basically these B cells, like I said, if we activate B cells in the lymph node and the spleen, they're just gonna produce and pump out. I mean, millions upon millions of billions of antibodies, and they're going to be secreted into the lymph and then into the bloodstream. But then those antibodies are going to get into tissue fluids. I mean, they're going to go all over the place. All right. So basically, what we're doing with this process is we are flooding our body fluids with antibodies that help us eliminate pathogens. All right. But remember, this is similar to cell-mediated immunity that we discussed in the previous video. That this is a form of specific immunity, and as a result of this, this is also a very adaptive process. Because remember, when we're thinking about the concept of adaptation, we are forming genetically specific memory cells to an antigen. So if we're ever exposed to it again, we can eradicate it much more quickly. All right, and essentially, when a naive B cell comes in contact with an antigen, whether it is through the contact of an antigen-presenting cell, or it's activated by a T helper cell, or it just comes in direct contact with the pathogen itself. They're going to basically undergo this clonal selection process and form into either plasma cells, which are the antibody-producing cells, or memory cells. Those already kind of, like I said, those genetically specific cells. Okay, so that's essentially that. So let's kind of talk about this activation process a little more. NB stands for naive B cell. This concept of clonal selection. So basically, what's going to happen now? B lymphocytes are a little different because B lymphocytes also have MHC2 molecules on their surface, but B lymphocytes also have an incredibly diverse array of receptors located on the surface of themselves as well. Now that wide array of receptors allows B cells to be activated by direct contact to foreign pathogens. All right, so that's something to keep in mind. Is that B cells can be activated by direct exposure to a pathogen itself, or more specifically to a foreign antigen itself. All right, and then typically what happens when those receptors come in contact with an antigen or a pathogen, this B cell will essentially phagocytize the pathogen and then start genetically processing it, and then undergoing that cloning or clonal selection process, and forming antibody-producing factories and memory cells. All right. Now, one thing to keep in mind about that, however, though, is that direct contact with the pathogens creates very, very minimal responses. All right, so the responses aren't really that great, or I should say vast, just by direct activation alone. the bulk of B cell activation is going to come from the assistance of T helper cells. Okay, it's going to come from the assistance of T helper cells. All right, so basically, T helper cells are basically, once they are activated, they are, like I said, they're going to release a bunch of chemicals, interleukins, cytokines, and so on, that are going to enhance the immune response, and part of that is increasing activation of B cells. Another thing T helper cells are going to do is they're basically going to remember, these T helper cells were activated by exposure to a pathogen from an antigen-presenting cell. These T helper cells are, in a similar fashion, going to take that antigen and show it to the MHC molecules of that B cell and turn it on. All right. And as a result, when you put the two together, all the chemicals being secreted and then the direct activation from the T helper cells, we have an exponential increase in B cell activity. Therefore, like I said, we're going to manufacture a lot more antibodies, and they're going to help us out a lot. I'll talk more about the specific functions of antibodies in a bit. All right. Now, one thing to bear in mind is that this process of basically forming clones and then converting it to different cells. Remember, when we're talking about a naive B cell, this is going to take a few days. This could take upwards of 4 days before these cells are getting to the point to where they are actually carrying out a specific immune function. So throughout that process, they're going to go through a series of divisions. All right, they're going to go through a series of divisions, and basically while they are doing that, the cells are basically genetically arranging themselves to that specific antigen that they were exposed to in the first place. All right, because remember, as I mentioned in the previous video, that the genes inside of these cells are a little different. I said that because remember, genes are found in different locations on a single strand of DNA. All right. Whereas with these B cells, these genes are kind of scattered all over the place throughout the throughout the nucleus. They're kind of found in clusters all over. And then basically what we do is we start splicing various regions of these genes and start smacking them together. So we form very specific codes. So we form very specific antibodies against that particular antigen. All right. So upon initial exposure to the pathogen, it's going to take a few days to basically genetically program these cells to do their job. And then remember, these memory cells are already, they already have the exact genetic code for that particular antigen. So when we're exposed to it again, they can immediately form right into the plasma cells that we need and produce and get the antibodies out that we really need. All right, immediately. Okay, so that's the job of a memory cell. Same thing with T cells. We talked about that in the last video. Now, plasma cells, these are the antibody factories. And I really didn't do this plasma cell a whole lot of justice. I really should have made this nucleus a lot bigger. But basically, there's a couple of key things to think about with these plasma cells. One of it is that they have a high volume of rough endoplasmic reticulum. Okay. They've got a high volume of rough endoplasmic reticulum, and they've got a lot of mitochondria. Okay. Now, let's kind of rewind here for a second. Remember, one of the most baseline concepts that you can ever remember related to, realistically anything biological, not just physiological, is that the sum of the functions of an organism are dictated by the cellular composition of that organism. Well, the same applies for cells. All right, the functionality of that cell is dictated by its composition of organelles. All right, so basically remember that the rough endoplasmic reticulum predominantly produces proteins that are going to be used for the purpose of export. Not all proteins, but most proteins produced on the rough ER are exported out of the cell. All right, so remember, the rough endoplasmic reticulum is an organelle. It's basically an extension of the nuclear membrane into the cytoplasm, And these green dots represent ribosomes, and ribosomes are the specific spots where we make proteins. All right, and obviously it's gonna, if we're gonna be making a lot of proteins here, we need a lot of mitochondria because, well, remember, making proteins is a very taxing process. All right, now once these plasma cells start kicking it in gear, they produce antibodies in high volumes. I mean, at a rate, realistically, it depends what book you read. Because, for example, Fox states that we produce about 2,000 antibodies per second. Guyton, if I remember correctly, says we produce antibodies at a rate of about 5,000 per second. Regardless of the numbers, that's an astronomical amount of antibodies being produced. So basically, these plasma cells then, for a day or two, are just going to sit there and just pump out, like I said, just in mass quantities, antibodies like crazy. All right, and then, like I said, these antibodies are going to get into the lymph, get into the blood, get into our body fluid compartments, and then these antibodies are going to interact with the antigens, and, like I said, carry out very, very specific actions. Now, something to keep in mind about B cells then is that they don't directly attack pathogens. The antibodies that they secrete essentially, they don't really even directly, I mean, in some cases they do, but in most cases, when you think about the functions of antibodies, they basically enhance other immune responses like phagocytosis, or they promote activity of various chemicals that kill and destroy cells and tissues. Or not cells and tissues, you idiot. The foreign pathogen that we're interacting with. Okay. Now, before we talk about the functions of antibodies, we need to just talk about the concept of essentially what they are. So the technical term for an antibody is immunoglobulin. Excuse me. That's pretty straightforward. We're talking about something related to the immune system. And globulin, that's basically, in the old days, that's essentially the word that was given. I mean, it still applies, in my opinion. But that's the word that's used to describe the chemical structure of proteins, because remember, proteins are essentially kind of twisted, specifically shaped. Don't get me wrong, they're very there's a lot of detail in the shape of a protein, but basically they are globular structures. They're essentially very complex globs of basically twisted amino acids that form, like I said, very globular but specific structures that carry out specific functions. But basically, when you see that word immunoglobulin, it should be a knee-jerk reflex to think that's what an antibody is. Okay, immunoglobulin equals antibody. Now, antibodies carry out a few different functions. One of them is neutralization, another one is complement fixation, agglutination, and then precipitation. All right, we're going to talk about these specific functions in a second. But first, let's talk about the structure of an antibody. So antibodies basically have two major regions. They've kind of got or a couple of major chains. This is what we refer to as the light chain, and then this larger in red here is what we refer to as the heavy chain. Okay, and now remember, just bear in mind that this is a very general picture of an antibody. I mean, the different classes of antibodies I'm going to talk about next come in different shapes, and that plays a big role in how they work. But in general, this is kind of what an antibody looks like if you were to kind of look for the common structures here. So basically, you notice that antibodies kind of form this letter Y, shape here. All right. Now basically, when we're thinking about the structure of these antibodies, there's a couple of things we need to think about. This heavy chain, especially this kind of lower part of the heavy chain, this is what I refer to as the base. Okay. And then the kind of you notice how this kind of looks like the letter Y. Basically, the ends of the letter Y, these are very important to focus on, because These are the parts of the antibody that actually interact with the antigen itself. Okay. Now, these are what are called the variable regions. Okay, the variable regions. All right, and if this was an immunology class, we'd be breaking this down more in depth because there's a variable region and then there's also a hypervariable region found within the end of these antibodies. But because this is AMP, we're just going to keep it more general. Now, the variable regions. Remember earlier when I talked about that selection process. You know, when these naive B cells are starting to divide and they're forming clones of themselves. And throughout this process, you know, throughout this couple days worth of division, with every cell division, all right, there's basically gene splicing and rearrangement going on. Okay. Now remember that antibodies are proteins, right? So basically, when this plasma cell is all done, all right, once we basically splice together the right combination of genes, you know, in the nucleus of these plasma cells, remember a gene is just a code for a specific protein, right? This variable region is going to be different with each antibody. All right, this is going to be a different combination of amino acids. It's going to be a slightly different structure for each type of antibody, and that's basically what gives us this vast array of diversity with antibodies and allows us to essentially interact with so many different antigens. And that's kind of, and a lot of that also comes from this concept of instead of genes kind of being isolated to specific regions on DNA, all right, by having all of these, you know, by having these kind of separated genes that we can kind of splice and rearrange and move around a lot more, that allows us to create a great amount of diversity with antibodies. Okay, so essentially, when an antibody is interacting with an antigen, it's going to look like this. Let's say this is a bacterium, and an antibody was actually interacting with it, you would look like this. Okay, so the variable region is what actually sticks to the antibody, and then this part that this base of the heavy chain, all right, kind of protrudes away from the antigen. Now that's important, however, because that relates to the functionality of this, which I'll talk about. Again, I'll go over those functions in a little bit. All right, but that's kind of your general structure of an antibody. Now, let's talk about the various classes of antibodies then. There are basically 5 different classes of immunoglobulins or antibodies that are associated, you know, with us that we use for defense purposes. One of those antibodies is and now remember, when you see this abbreviation here, IG stands for immunoglobulins. Okay, immunoglobulins. Now, these antibodies vary in function essentially because they vary in structure. Okay, so and again, because this is anatomy and not immunology, we're not going to cover the different structures of these, but just bear in mind that these are all very, very different antibodies, okay? But they still interact with antigens per se. So immunoglobulin A, IgA, basically this is an antibody that is found in bodily secretions, okay? Bodily secretions. Now that's important because, remember, bodily secretions are essentially, these are essentially materials that are being moved to the outer surfaces of the body. In a lot of areas where there are secretions, the inside of the body is directly exposed to the outside. Like, for example, the mucous membranes. Okay, the mucous membranes. Mucus is a secretion produced by goblet cells, all right? So we can mix some IgA in there and think about that. Most bacteria have flagella that are used to propel and move them around. All right, so if we start sticking some of these antibodies to the flagella of these bacteria, that's essentially going to neutralize or immobilize them and prevent them from moving around. And then the lytic enzymes in the mucus can essentially, it makes it easier for the enzyme to interact with the bacteria if we kind of make it stationary, and we can kill it off. All right, so that's an example of a secretion. Think of what are some, pause for a sec. and try to write down as many secretions that you can think of here. I'll give you a second to do that. All right, so let's think of some other examples of secretions where we would find just secretions in general. Sweat, tears. Here, got mucus out of the way. The oils. Let's see what else here. Cerumen, which is essentially earwax. Let's see here. I got the mucus out of the way. So we're missing one. Sweat, tears. Here's one that students like to say, but it's incorrect. Urine. Urine is an excretion. Urine is an excretion. Okay. Now, secretions are materials that are produced by glands, and then they're secreted from the glands. All right. And a secretion is typically some kind of water or oil-based material that is moving from one body surface to another, the inner surface of the gland to another body surface. And a lot of bodily secretions are secreted, especially from exocrine glands, are secreted onto the surface of the body, skin. All right, not all, but most secretion, the exocrine glands. Now, urine, however, remember, urine is filtered blood. All right, we filter water and small materials out of blood, and then we process it, and then we eliminate it from the body. However, there isn't urine. All right. There should be practically no protein in your urine. Okay, if there is, that's going to be a sign that there's probably something wrong with your kidneys. All right, so that's something to keep in mind. All right, so urine doesn't fall into that. So in general, immunoglobulin A found in bodily secretions, and is like I said, is a good protector against, like I said, materials that come in contact with body surfaces like bacteria, fungi, and so on. All right. Immunoglobulin E, IgE, is a kind of a player in allergies, and that's essentially what a hypersensitivity reaction is. That's kind of the more technical term for an allergic reaction. That's one type of hypersensitivity anyway. Basically, when we're thinking of allergies, like hay fever or hives and things like that, allergic reactions are when a B cell comes in contact with what we call an allergen. Okay, an allergen could realistically be anything. It could range from dander from an animal. It could be. It could be metal. It could. Like if you wear, let's say, I don't know. You develop an allergy to copper because you wear you wear a copper bracelet and never take it off or whatnot. So some kind of molecule in food. All right, but basically, what can happen if B cells come in contact with an allergen, what they can do is they can produce IgE. All right, then what IgE is going to do is it's going to bind to mast cells. And basically, here's how the antibody. So basically, this antibody IgE is going to act as a receptor on these mast cells. Now, when IgE interacts with these mast cells, we would say that they are sensitized. All right, that they are sensitized. Now, basically, so then what happens then when we're exposed to that allergen again, and that allergen comes in contact with these antibody receptors on these mast cells, that's going to cause these mast cells to degranulate and dump out histamine. Now, remember where mast cells position themselves. They're in many cases in the body, they're found hovering around capillary beds, or also around. They're found in abundance in the connective tissues around your bronchioles, your microscopic airways in your lungs. That's why typically, when someone has an allergic reaction, the responses are almost always identical. They're very similar. Just depends on how severe the reaction is. But basically, you're going to see some kind of swelling or inflammation because histamine, remember, increases capillary permeability, and um, and then it can also cause respiratory complications if because histamine causes your bronchioles to constrict. That's why when someone has a severe allergic reaction, they give them, they take epinephrine because epinephrine counteracts and antagonizes the effects of histamine and dilates your bronchioles versus constricting them. All right, but IgE, like I said, is primarily involved in, like I said, sensitizing mast cells and promoting allergic reactions. It can also be used in battling parasites as well, but we'll just, for the sake of discussion, we'll primarily associate it with allergic reactions. That's the other one I was thinking of. I'm sorry. Let's rewind here for a second. So I'm done with IgE, by the way. I completely forgot to mention breast milk for IgA. Okay, breast milk. Good Lord, how did I forget that? Now remember, this is, and the reason why I'm coming back to this, I apologize. The reason why I'm coming back to this is because that's what happens when you do this at 2 in the morning. Yeah. So basically, when a baby's first born, its digestive system isn't fully developed yet, and actually it has the digestive system is very porous. All right. So basically, what happens then? This is one of the few proteins that's actually found in breast milk. Remember, breast milk is predominantly water and fat. And like I said, one of the main proteins in there is this antibody IgA. So basically, because of the kind of the more kind of, again, quote unquote porous nature of the kind of the neonate's GI tract, they can absorb these antibodies into the bloodstream, and then that basically helps them slowly build up their immune system as well. Now, if a kid doesn't breastfeed, don't get me wrong, they're not going to have kind of some bunk immune system. It's just that this just kind of helps. All right. So I forgot to mention that, sorry. IgM and IgG, these are always kind of the more confusing ones because they're similar in function. But basically IgM, kind of the key thing that's associated with this is that when we say primary response, basically what we're saying is when we first come in contact with an antigen, the first time we see it and react to it, this is the antibody that we secrete in high amounts. Okay, this is the antibody that we secrete in high amounts. And one of the things one of the functions that this induces is something called complement fixation, the activation of proteins in our blood that basically cause the destruction of cells, which we'll talk about more in detail in a minute, but it's actually kind of cool. Another thing that IgM can create is agglutination. Agglutination basically that's the high-class word for clumping. Okay, that's the high-class word for clumping. Now, so basically what this does is, I'll come back to that in a little bit when I talk about the functions. But clumping, basically, but that's essentially, so this kind of, for now, just think of that clumping. So the main thing I guess I really want you to think about is complement fixation and the primary response. Whereas IgG, notice similar reactions here: complement fixation, neutralization. All right, helps enhance phagocytosis. A big one here, though, is that this is actually the most abundant antibody in our blood in the first place, and it plays a very large role in the secondary response. So when we're exposed to an antigen the second time around, and then we activate those memory cells, all right, immunoglobulin G is the antibody that we're going to primarily secrete. Okay. Now, another key factor with IgG, which I can't believe I didn't write that down or put that on here, is that it can cross the placenta. Okay, it can cross the placenta. So basically, what I'm saying is that this antibody can move from mother to child, or mother to the embryo, I should say. All right, now that's important because, for example, during embryonic development, this is part, it's slow, but this is part of the development of the immune system by acquiring these antibodies. by passively, naturally acquiring antibodies. Um, and another thing with this, for example, like if a mother, if you're talking about blood typing, if mom is Rh negative and baby is Rh positive, that could, I'm not guaranteeing this is going to happen, but this could potentially cause a problem because remember, if mom is Rh negative, that's a molecule on erythrocytes that mom has never seen before. All right, so if baby has this particular antigen or marker on its erythrocytes, well, guess what can happen? That could elicit a response. And then typically, if this does happen, what will happen is that the first baby will be fine, but you run the risk of developing antibodies against that Rh factor. And then what can happen is if mom gets pregnant again and there's another baby that's Rh positive, there's a chance that those antibodies can react to those erythrocytes and cause the destruction of the erythrocytes, which will cause lots of damage to the child. All right, so that's another key factor with IgG is that it can cross the placenta from mom to baby. And then IgD, this is certainly the most poorly understood of all of the antibodies. Basically, the best explanation that can be given is that it's found on the surface of B cells, but realistically, its exact role is poorly understood at best, even to this day. So, I really honestly don't have a whole lot to say about IgD. Yeah, I don't know. If only everything was that easy, right? So basically, in a nutshell, that's kind of the functions of the major classes of antibodies. Now let's kind of go over some of these functions that were mentioned before, or some of these words: complement fixation, agglutination, neutralization, all that stuff. So neutralization, antibodies can neutralize the activity of certain molecules or pathogens just by them binding to to the pathogens themselves or to various molecules. For example, neutralizing certain toxins that bacteria secrete. Like for example, when you get your tetanus vaccine, what you're doing is you're not getting the tetanus vaccine against the bacterium itself, Mycobacterium tetanae. If I remember correctly, that's what it's called. But you're getting the vaccine against a particular toxin that it secretes that disrupts, that basically kind of disrupts the neuromuscular junction and acetylcholine receptor activity and so on, and causes a very severe and potentially life-threatening muscle spasms. So what you're doing is you're developing antibodies. What you do when you get that shot is you are going to develop antibodies against that toxin. So if you're ever exposed to it, you've already got those antibodies in your blood, and then they are going to basically, if you come in contact with that toxin, they're just going to stick to it and neutralize it from interacting with our receptors and causing harm. All right, like antivenoms are another good example of that. Basically, like with an antivenom, what they'll do is they'll take a venom and they'll put a little bit of it in like an animal, and then they'll extract the antibodies, out of that animal for that venom, and then they'll, they'll bottle it up. And if you ever get bit by a snake or a venomous animal, then you get the antibody, and then the antibody prevents those, basically the venom or the toxic aspects of the venom from damaging you. Because remember, venoms essentially mimic, in a lot of cases, chemical receptors. And like for example, neurotoxins bind to, a lot of them shut down your respiratory center by binding to receptors on neurons that shut down your respiratory center. Some of them can cause muscle spasms and cause paralysis and breathing. Some of them are hemolytic and cause destruction of red blood cells. In most cases, you're probably not going to get to the doctor if you get that venom in you, because those are it's incredibly lethal. But you get the picture here of neutralization. Another function of neutralization, like I already mentioned this when I was talking about binding to, for example, flagella on bacteria, because remember, that's how bacteria move around. They kind of They kind of roll around and then they skate around and they tumble and then they move around. They do so with these flagella. But if we get a bunch of antibodies basically sticking to flagella and weighing it down and preventing it from bacteria from moving around, we neutralize that bacteria's ability to essentially relocate itself. And then basically by immobilizing it, it's a lot easier to kill it. It's a lot easier for a phagocyte to kill a stationary pathogen than one that's moving around. So that essentially is neutralization, is neutralizing the function of something. Agglutination and precipitation, basically, what we're saying here with agglutination, like I said, that's the term for clumping. All right, remember we said that IgM is a big player in this. So basically, now what you're noticing here is that these circles are supposed to represent IgM, and notice how one IgM antibody can bind to multiple multiple cells at a time. In red here, these are supposed to represent RBCs or erythrocytes. So basically, what happens here? I mean, this is essentially a transfusion reaction. If you're given the wrong blood type, the wrong blood type, then what can happen is those antibodies that are already in your blood are going to immediately bind to these foreign red blood cells and then cause them to clump together. Now picture this trying to move through the microcirculation, not going to be good. That's why when people get transfusion reactions, they pretty much notice immediately. When I say immediately, I mean within like seconds to minutes. Because these agglutinated red blood cells, again, they obstruct blood flow and can cause ischemia. Typically, like I said, because we catch these so quickly, and they're just rare anyway, transfusion reactions, they're typically very, very manageable, but if left alone, they could cause severe damage to the kidneys, by getting these big clumps of blood cells stuck in small vessels and causing damage. So. And then precipitation, basically, if we clump a bunch of cells or molecules together, that essentially is going to weigh them down. I mean, that's like the equivalent of going all mafia style and tying a cinder block to somebody's feet and then throwing them into a lake, and they're being weighed down and they sink to the bottom. All right, these antibodies are protein molecules. All right, so when you get these proteins sticking to these pathogens, that's going to weigh them down and then make it easier for, again, phagocytes to come around and, eat them up, right? Consume the unwanted material. All right. So that's how they can essentially enhance phagocytosis as well. The other main function that I really want to talk about was called complement fixation. In our blood right now, we have these plasma proteins that are called complement proteins, and right now they're in an inactive state. Now, when antibodies bind to antigens, what happens then is we start activating complement proteins, and this is a really, really specific process here. I mean, there's a specific. Again, I'm not going into the nitty-gritty detail about this, but basically, there's a specific complement protein that kicks off this process. There's specific enzymes involved in this process, but this is kind of a cascade of events. But basically, remember before when I was talking about the antibodies, and I said that the variable region is what actually interacts with the antigen, and then this base of the heavy chain kind of protrudes away from the pathogen. Now, the complement proteins, what they do is they interact with that heavy chain, that heavy chain. They start sticking to them, and then that basically is what activates this cascade of chemical reactions. We activate complement. All right, and then basically once we start activating complement, there's a series of, complement can enhance inflammation, complement enhances phagocytosis, and complement also directly kills things as well. So basically what I have here is basically what complement proteins do is when it comes to killing a pathogen, like for example a bacterium, what they're going to do is these complement proteins are going to stick together and form this circular unit, which is essentially a hole punch. And then basically they're going to pop holes. They're going to break open the plasma membrane. They're literally just going to penetrate the right through the plasma membrane, and that's going to cause an influx of fluids. It'll cause some fluids and materials to leak out as well, but that'll cause essentially the cell, to kind of swell up and then rupture and then die. And then that essentially, again, enhances phagocytosis. Some of these complement proteins essentially tagging these, these foreign invaders, causing their destruction, and then phagocytes cleaning the mess. All right, and then over here, like I said, this is enhancing inflammation. Repeat inflammation. Complement proteins, like I said, they can enhance, white blood cell migration to the tissues, mast cell, granted, you name it. They do a lot of things, but in general, they also enhance inflammation. Now, that's important because when you think of autoimmune diseases, that's part of the process of why some autoimmune diseases behave the way they do, because we can produce autoantibodies, auto means self, antibodies against self-antigens that essentially will cause, that will activate complement, except complement will attack our own tissues instead of foreign tissues. Now, after basically covering all of that material, not just with B cells, but with T cells in general, there are certain ways we develop our immune system, and realistically, the main way we develop our immune system is through exposure. All right. So basically, in general, there are two ways to do this: active and passive. Basically, when we develop our immune system through active means, this is basically exposure to antigens. All right. Whereas with passive immunity, this is where we have outside sources that help us acquire materials to help to enhance the immune response. All right. So, for example, basically, we can through active immunity, the easiest thing to think of is just coming in contact with a pathogen. Getting sick, just naturally getting sick. You get the seasonal flu. And realistically, I'm a firm believer that unless you're a child or an elderly person, you don't need to listen to the scare tactics. Don't get me wrong, I got nothing against the flu vaccine, but I think they push it a little too hard on people. Realistically, just get the gallbladder thing and get sick. And if you get sick enough to the point where there's clinical problems, then go to the hospital. But otherwise, wait it out like most other people have to do. But basically, what's going to happen is, when you get sick, you see that antigen, you start to, you develop those memory cells, and then when you encounter that antigen again, you can just eradicate it really quickly. All right, and so you can develop memory by just active exposure, just direct exposure to pathogens, or also by indirect exposure to pathogens. Indirect might not be the best word, but basically by let's just say it, vaccines. Okay, vaccines. Because basically the premise behind a vaccine is that you are, like, let's say for example a vaccine against a virus. What they'll do is they'll take the virus and they'll one of the more common things that they do is they'll infect an egg, like an actual chicken egg. They'll infect the cells in there, let the virus replicate, then they'll extrapolate all the different variations of the viruses out of there, and then they'll basically kill the virus. They'll mix it in some chemicals, blend it up, and then damage it. But what's left is, but the virus doesn't have to be alive to react to it. The thing you need to see are the antigens on its surface. All right. So then what they do is they take that virus and then they inject it into you. You see those antigens and then you attack. All right. Problem is not every single one of those viruses is completely rendered inactive. When you're trying to attenuate it or process it for the process of the vaccine, you got to remember, you're coming in contact with an antigen itself. You're going to get sick. All right, you might get sick. You might carry out an immune response. All right, so that's something to keep in mind about that. And but you see that the theme here is that you're basically promoting whether you're just actually getting sick or you're coming in contact with an attenuated virus in a vaccine. You have direct exposure to the antigens on that material, and therefore you're developing memory cells and antibodies against that pathogen. So whenever you're naturally exposed to it again, you're ready for it. Okay, you're essentially ready for it. And I really don't want to hear anybody talking about that whole vaccine and autism hoopla, because that's all it is, it's hoopla. So, yeah, I just don't feel like touching out a 10-foot pole in this one, because that's just not true. Now, with passive immunity, like I said, this is where you basically acquire antibodies or other chemicals through outside sources. And again, you can do that artificially or naturally, just like with active exposure, natural exposure to a pathogen or artificial exposure. With passive immunity, this is where, for example, they can make monoclonal antibodies. Basically where they like take a mouse, expose it to a pathogen, and then that mouse, and then you take the B cells out of that mouse, kind of combine it with some tumor cells, and then you start to form specific antibodies against a particular pathogen. And then you can take those antibodies and inject them into someone if they're infected with a particular pathogen, and then those antibodies help kill whatever is affecting that person. All right. Or other chemicals like if you work in a hospital or clinical setting, you might hear orders for IV interferon. Interferon, remember, is used to enhance our defense against viral infections and make it easier for the immune system to attack viruses. By enhancing fever, phagocytosis, and so on. So, but that's all that. But that's what passive is like I said. Like I gave the example earlier, breastfeeding as well. If you're taking in mother's milk and then you're acquiring IgA, or you're passively acquiring IgG as it's crossing the placenta as you're undergoing embryonic development. All right. But that's essentially, but in a nutshell, realistically, you develop your immune system by exposure. You get sick. All right. But in some cases, you might need assistance, and we might need to have outside sources, again, where we need to put antibodies in ourselves or have other chemicals put in us to help us fight off the pathogen. All right, I apologize for the density and the length of this video, but there's a lot to cover in this. The last thing that I want to talk about is, in another video, is basically just what can happen when the immune system goes wrong. So basically immunodeficiency and autoimmunity.